our own, part of our Drexel family. Today is going to be the Andrea Messina Memorial Award, and after our guest speaker today, we will have the announcement of the award winner. I want to welcome the Messina family, and we're so happy to have them here. We also have a number of family and friends that are joining us by web stream, so that we welcome them as well. When we were thinking about this award, we wanted to think of a speaker that would represent the award, but also someone that would have meaning to us, to you, and one of the perfect people that came to mind was Dean Marasco. For your time here, she has always been your dean, but she actually is going to be stepping down from that position this summer before the start of fall. So she has been dean since 2003, and so most of my time here, she has also been here. Although she was not originally part of the Drexel main campus, she was down at the former MCP Hahnemann, and she was there as the chair of the microbiology and immunology department there before she came down to main campus to take over the stewardship of the College of Arts and Sciences. The dean has been a heavy hitter in the sciences for most of her time, and especially for the young ladies in the audience, I don't know if you appreciate the fact of once upon a time, the number of women in sciences was very, very small. I won't give my age, but back in the day, when I was doing my undergraduate, I really was in a large department, and there was only 2 female faculty on staff. And so she is from a time where she was definitely a pioneer of women in sciences, and she has always maintained that as one of her passions throughout her time, both when she was down at Drexel Med and also in the College of Arts and Sciences. She got her degree from Douglas College of Rutgers in bacteriology. So she is a microbiologist by heart and training, but she then went to the Hershey Medical School where she did her PhD in microbiology. So she then went for postdoctoral work, first to the Institute of Cancer Research, and then a stint at the Jackson Laboratories in Bar Harbor. She has always been interested in microbiology, but her most recent work that I know is her work with the influenza and the flu. And she's been doing this so that once upon a time I was actually a participant in one of her studies back when I was able to be in the under 40 category. So we start to definitely see the fact that she has been a leader within the College of Arts and Sciences, and as she steps down from the deanship, she will actually be a faculty member within the department. So she is one of our own, so we are very excited to have her back in the department when she finally officially turns over the reins. But I knew that she would be a perfect speaker for today because Andrea would have been someone that she was probably going to be her biggest cheerleader and the person behind motivating people to follow their dreams. And so without further ado, I'd like to introduce the dean and have her tell you about the work that she's been doing throughout her career. So thank you.
Knowing that this talk today was because of Andrea, I wanted to talk not my normal talk. I'm going to go into incredible detail about the science and how everything is done. But what you need to be as a scientist is have the ability to change, to think, to incorporate, but to find so many things interesting. And the one thing that everyone knew about Andrea was that everything was interesting to her. And everything about biology and science and veterinary medicine was going to excite her forever. And I hope you get from this talk that I've been excited about what I've been doing. Seems like forever. So I entitled it Viral Immunology. So yes, my undergraduate degree was in bacteriology, but I actually went to graduate school to be an immunologist. But I decided I wanted to do viruses, because back eons ago, we thought that everything was known and every bacteria was fine. We didn't talk about resistance of bacteria at that time. We hadn't been so overly using antibiotics that we created these monsters that we deal with today. So the real area was we hadn't done enough with vaccines. So that's why I really wanted to go into viral immunology initially. But it was also the early 1970s, and at that time, President Nixon declared a war on cancer. And there was a big thought or a push within the cancer community that all cancers were going to be caused by viruses. So that sounded like a pretty good place for me to start. So when I was at the Hershey Medical Center, I had the auspicious title of my thesis called Demonstration of Virus-Associated Membrane Antigens on Hamster Cells Transformed by Cytomegalovirus. Like, working with hamsters is not very much fun, by the way. But if you look today, cytomegalovirus is not a major player in cancer, maybe a minor player. But the department I joined really felt that all herpes viruses, and cytomegalovirus is a herpes virus, were going to cause cancer because they have the ability to stay in your body. You get infected with a herpes virus and they never go away. So what a perfect agent to do something to a cell since it already has the ability to hide. So it seemed to be a pretty good model that I'd work on. And so a graduate student before me had actually taken hamster embryonic fibroblasts and treated them with virus in a tissue culture dish and got some that would grow in hamsters. And then I got them and said, gee, do these have antigens that we can deal with? This is actually a page from my thesis. You can see the terrible type. There was no such thing as computers. There was no such thing as correcting right away. All at end for my thesis, I had to give an error-free document to Penn State, which meant I had to hire a typist because there was no way it was going to happen. But what this slide is very complex, but I just wanted to show you, something directly from my thesis is that, whoops, a little button here. These were cells that were transformed by cytomegalovirus, and when we put them into hamsters, they grew. And we then would take, and there are three, actually there are about 7 lines, but 3 that we used all the time. And we wanted to say whether or not, if we take spleen cells from it, whether or not these cells will die. You now, in any course you've taken, you've heard about cytotoxic T lymphocytes. Let you know the first time that anyone demonstrated that was in 1969, and I started my graduate work in 1972. So people were still questioning whether or not there was such a thing as cytotoxic T lymphocytes, and more importantly, can it do anything to a tumor cell? I'll show you the assay in a minute. You won't believe what we did, but basically what you found is you took spleen cells from a hamster that had a tumor. You then used the transformed lines as targets, and you would put them with normal spleen cells or immune spleen cells. Now, mind you, these were full spleen cells, didn't divide anything because we didn't know how to yet. Okay? And what you found is that we would have killing of the cells. So we had cytotoxicity of 20 to 50%, and if we just put these cells on, if we put the spleen cells with normal fibroblasts, they didn't do anything. Good thing it worked, that was my thesis. But this is how it's done. This is a petri dish. The red dots represent the tumor cells. The small dots represent the lymphocytes. 96-well plates, you've seen them sometimes. You now have smaller ones. We would plate them in there, and I would sit over a microscope and count them with the clicker, one by one. There was no way to count them. There was no computer to count them. There was nothing. You would just have to sit and count. And you would score, learn how to score the bottom with a grid underneath so they wouldn't count something twice. So you sat there going blind. So it was a lot of work. Over time, things got a lot better, which is what we're going to be talking about. But that's how it all started, and that was the basis of my graduate work. Now, this was pretty big because a lot of people were saying that there were no antigens on tumor cells. Right now, if anyone says there's no antigen on cancer cells, I say, what? You know, we're having this whole immunorevolution right now. But you'll see at the end of my talk, I actually stopped doing cancer research because no one wanted to fund us, because people weren't believing that this was going to work. Those of us who are in the field thought so from the beginning. We would go to meetings back in the early 70s to about till the 80s, and we'd go to sessions that had a room this big and we'd have 20 people in it going to listen to it. I went to a cancer meeting about 2-3 years ago in a room that held 1,500. It was standing room only. So if you really feel that what you're working on makes sense, try to stay in it because it may work someday. So then, although I was at the Institute for Cancer Research in Fox Chase, my postdoctoral advisor became director of the Jackson Laboratories. And one of the things we knew at the time that in mice, there were actually viruses that really did cause cancer. There were the leukemia viruses. So I went to the Jackson Labs, which is the mouse mecca, all inbred strains of mice, to learn how to work with them appropriately. And in addition, to see if there's such a thing as vaccination against these murine leukemia viruses. So the model, the tumor that we're using was a virus called Maloney sarcoma virus. We had cells transformed by it. And what I did is, after playing with the model, figuring out how many cells I needed to cause a tumor, how long the animals died and everything, what I did is purify the virus the best we had, which was basically by ultracentrifugation, and that's it, and just gradient cuts. And using as purified as we can, immunize with varying doses of virus. And the interesting thing is, if you just gave the virus by itself, what you had was the 55% of the animals came down with tumors. If you gave a high dose of virus, you cut that in half. So you immunized with 10 to the 10 particles, and you got half. But the weird thing that was happening was that if we immunized with low doses of virus, we actually got an increase in tumor. And at the time, again, this is around 1977, 78, there's a whole theory called immunostimulation, which also fell out of favor, because we didn't know about such things as cytokinetics. And it turns out that long after we looked at this, that this particular tumor has a receptor for what we now call interleukin-6. And if the small amount of virus given early actually induces interleukin-6, and it actually acts as a growth factor on these particular tumor cells. And when you went higher, the immune response generated to the virus overcame the amount of small amounts of cytokine which act as a growth factor. But we had no idea what was happening, because when I did this study, we didn't even know there was such a thing as IL-2, which was necessary for the growth of T cells. That actually came out, was presented at a poster presentation. Let's say I was in Bar Harbor in 76, came out in 77 as a presentation at a meeting. That you had these things in fluid of cells that you put in culture that would make other cells grow. And it was called just T cell growth factor. It wasn't categorized yet. But it was saying, okay, we really do have something that we can do here. Let's try to work with this sometime. But I finished my postdoc, and I became a faculty member at the Medical College of Pennsylvania, which has also been known as MCP Hahnemann, which is now known as the Drexel College of Medicine. And I continued my work there, and trying to, at the same time, because of this whole area of cancer research, a factor came out interferon. Interferon is something that is produced by a cell when it's infected by a virus. So the virus comes in, and with most viruses, the cells induce interferon within 3 to 9 hours after the virus comes in. The cell that gets infected dies, but the substance interferon goes out, attaches to the cells in the area, and those are protected. But a group that started first in Sweden said they took this substance out and put it on tumor cells and they stopped growing. And they used a chemically induced tumor and the tumor didn't grow. So they were proposing that interferon could be used as an anti-cancer agent. And indeed, it does work in limited amounts. Problem is, it does a lot of other nasty things, which I also investigated, but we're not going to go into today. But since I was interested in the murine leukemia viruses, I wanted to see whether or not the viruses induced interferon. Because if they did, wouldn't that kind of limit the infection? So these two viruses, Gross and Friend, are viruses that were isolated from animals at some time, and we grew them in tissue culture. And then we would take this tissue culture with this little bit of purification and put them into animals and induce tumors. The bottom virus, AKR, is a virus that I just isolated from an animal with leukemia. The AKR strain of mice just spontaneously get leukemia. And by the time these animals are 11 months old, they're already dead from leukemia. And the leukemia is caused by a virus. So I said, okay, let's look at these 3 viruses. Well, the ones that we grow indeed induced interferon. The one that I isolated freshly from the animal did not induce interferon. So the question is, like, what was happening? Did we have a contaminant in here that was causing interferon? We tried every way to purify it out, and we couldn't find anything except that this fresh AKR virus did not induce interferon at all. At the end, I'll also go through different assays, and this was a bioassay. So if we're doing assays now for something like cytokines or interferon, we do ELISAs. It's easy, quick little assay. This was an assay where we actually had to treat cells with interferon, wait for 24 hours, infect the virus, wait for 24 hours, and see how many of the cells that were infected died. Again, counting under the microscope. Lots of fun. That's all we had. So looking at this, I wanted to see, since these animals died at 11 months, whether or not if we did something early when the animal was developing, whether or not we gave them interferon, whether or not we can limit the development of the leukemia. If interferon works, maybe we should do it early. We knew it was not transmitted from the mother. It was in the DNA. We now know, because we know now that retroviruses incorporate into the DNA. So what I did is, for 7 days before we expected the pups to be born, I gave them interferon. And then when the pups were born, checked them for the amount of virus that they had. And what we found is, of the 9 pups on day 1, only 1 had virus. And on day 7, only 1 of 8 had virus. But by 1 month, they all had virus. Now, they were not treated after birth. They were just treated the week before birth. If we gave mock interferon, which is basically inducing it the same way but not having the final inducing agent, everyone had virus. And if we just gave saline to the animals, they had virus. So it seemed if we gave interferon in utero, we'd be able to stop it, but not permanently. I've never pursued the mechanism. I can't tell you what was happening in utero, but it was very reproducible. And you can see the titers are about the same. By a month, they're all about 10 to the 4 units per gram of tissue. And before that, it's only 100 units. Now, in order to do the interferon research, I just said we just gave them interferon. What would we do today? We'd go buy it. Couldn't buy it back in the day. So I had to produce myself the the interferon that I was going to use, and I had to purify it. And part of the purification was developing that we ultimately used. Initially, it was just by size and charge, but eventually what we did is developed a polyclonal antibody in sheep. Had to grow it up, grow up all the contaminants, and go out to New Bolton and immunize sheep with interferon. And we developed this polyclonal antibody, and we would bind the antibody to a column, throw the prep that we're making through the column, the interferon would stay, the rest of the garbage would go through, and then we do some chemical, a biochemical characterization. It was clean, but it wasn't real clean, because polyclonal antibodies aren't specific, but there was no such thing as monoclonal antibodies until 1980 when they were developed. So once that whole technology of developing monoclonal antibodies came out, my lab and I started to make a monoclonal antibody against the interferon so we can have pure interferon. And for a while we actually produced the monoclonal antibody that was used throughout the United States to purify interferon until some companies played with it more and had a higher affinity for the interferon. But just as an aside to do what you don't have a reagent, you make it. And at the same time that the monoclonals were coming up, someone said, ooh, now we have really good antibodies. Let's have a better assay. So at the same time, the ELISA assays were coming up. So this is where you have an enzyme-linked reagent, and if the antibody binds to the antigen, you get a change in color. And then all you have to do is put it through a spec to find out if it works or not. But that all was happening at the same time, and we also had to develop the whole ELISA. We wouldn't even think of it now, but the thing is, it's not real consistent with change in humidity. The whole reaction is totally messed up if it's too humid or too dry. So now you know how to deal with it, but back in the day it was not. So I now have this interferon. I go back to the tumor system that I had, that I showed you earlier, and I now, in an adult animal, 6, 8 weeks old, adult enough, we gave interferon 4 days after this is not the virus, this is a tumor transformed by the virus. If we give the tumor and we'd give it in the thigh, the tumor would grow, and a certain percentage of them would metastasize to the lung. If we gave interferon for 4 days after the tumor, our incidence was only 28%. Whereas if we only gave saline, we had 100% tumor incidence. Metastasis was about 50%, 44% normally, and we cut it down to 3%. This worked, but the thing is it only worked when the tumor cells were given in that short interval, that the interferon was so close to the tumor. So if you already had a tumor, we had no effect. We can only do it as we're giving the cells, so it really had no human application at that point in time. But go back to immunology. So I'm just giving stuff here. We didn't know how it's working. So the MSC is the abbreviation for the cells we used. And we now took out spleen cells, and we mixed them with the tumor cells, and we now want to see how many cells we killed. By this time, we had another assay. So I didn't have to count these visually anymore. And that's a good thing because these cells didn't attach to wells as well as my first one, which allowed me to do the assay. Here we labeled the tumor cells with chromium-51, a radioactive component that goes into the cytoplasm of cells. And after a certain number of hours, and that depended on the cell type that you had to standardize, you can see how much of the radioactivity was released into the supername. So you can see what cytotox was. And what we found in the animals that we had given interferon to, we now got cytotoxicity. Whereas the animals, the circles are animals who had no tumors, and the squares down here are animals who had the tumor but no interferon, no cytotoxicity. So this was one of the early demonstrations that interferon had the ability to help an immune response occur. We now know that gamma interferon comes up after, but this was the alpha beta interferon. I told you a couple minutes ago, alpha beta interferon comes up very early after a viral infection. It works against the virus, but it also increases the immune response to the virus. And it has the ability to do the same thing here when the antigen is introduced into the system. But there's a little bit of a problem. I told you a while ago that when we did our first cells, we had no markers to tell what it was. It was a spleen cell. Okay, so we can tell by its morphology, it's a lymphocyte. We didn't know initially. We did know it was a B cell if it had antibody on it, if it had immune globulin on the surface. But our markers for T cells are really terrible. By the time I was doing this, I told you we had monoclonal antibodies. We now had monoclonal antibodies that will identify our subsets of T cells. We also had a monoclonal antibody to identify this other cell that was a lymphocyte, but it didn't bind what the T cell was. And we called it a natural killer cell. Why? Because regardless of what we did, it was always there. So it killed cells at a low level, and we didn't have to do anything to get it there. So just to the slide that I just told you about, here's the tumor. We gave the interferon, and before we mix these splenocytes with the tumor cells, we either treated it with the anti-T cell antibody or the anti-NK cell antibody. So we have about 21% cytotoxicity. If we give the anti-T cell, we reduce it a bit, but wipe out almost all of it with the anti-NK cell antibody. So what we were finding was that when we gave interferon in the system, while it has the ability to enhance a specific immune response, what it does far, far better is induce NK cells to kill. So while they normally have a background level of killing, you give them interferon and they're really ready to kill. Why is that great? Because these kill viral infected cells really well. So while it takes 7 to 10 days to generate a specific T cell response to a virus, the interferon comes in and gets those NK cells to cut down the viral load early. And you could do it with a tumor that has a virus on it as well. So I was happily doing my cancer research. Grants were getting a little bit harder to get, but I was still getting them. And I'm in College of Medicine. And the chair of the Department of Medicine calls me to his office. You always go if the chair of medicine calls, like a really powerful man in medical school. And obviously a man, because we had no women chairs of departments when I was there at all. So I go. And he said, I understand you're an immunologist. And I said, yeah. And he said, well, I have a project that I want you to be on. We want to do work with aging. And I read that the immune system decreases with age. I want you to test the people that we're recruiting into our studies. And I said, I don't do people. I do animals. And he said, oh, it can't be that hard. Which is indeed not true. Inbred animals have far less variation in the responses you get than the heterogeneity of people. But again, here was someone saying, I'll give you some startup funds, you can start something new. So you figured, why not? So I did. And so the first thing he just wanted me to do is, how does the immune system work in the elderly? He doesn't care if it's a virus, he doesn't care if anything, just check it. So the easiest assay to do is to throw in a stimulus with the cells, the lymphocytes that you get from the blood, and see if they proliferate to the stimulus, which are called mitogens. And so in this slide, this is the age across span. Each dot represents about 40 people. And the yellow line are women, and the red line is men. So there's about 80 people at every dot here across the different deciles. And what you can see when you put this mitogenic stimulus in, that the immune system goes down to this mitogen, which is a T cell mitogen, this mitogen, which is a T cell mitogen, this is spiking hemagglutinin and concanavalin A, and this is protein mitogen, which stimulates B cells. So B cells don't change a heck of a lot, but the T cells do. And the interesting thing, if you look here, that you kind of level out when you get in the 70s and beyond. It's a little more interesting than that. So he was excited. Now we have this marker, but this is what you really see with the dots. That the dots are all over the place. And so you will have an individual here who looks the same as a 20-year-old. But what you don't have in this age group are the really low responses. So this heterogeneity is what excited me more than kind of the mean responses, because if we're trying to map this to getting sick with urinary tract infections, development of cancer, whatever, I need to look at the heterogeneity. And so we have definitions that we made that a low elderly responder had less than 20% of the mean response of young, a high was greater than 60, and the mid was everything in between. And young, as Dr. Jules said, all of our studies use young who were less than 40, okay? And you didn't get tested at all in our studies if you're between 40 and 65. So that was when you're free from being bled in the lab, okay? But once you got 65, it was fair game. And so what we would find generally is that when we used one of our T-cell mitogens, the young, this was by treated thymidine incorporation for proliferation. Easiest thing at the time. You don't like to use it a lot now, because why? You don't want to get rid of radioactivity. Not a good thing. Back in the day, it's easy. So you throw in, look at the counts of treated thymidine that's in there, because as the cell is replicating, the labeled thymidine is in there and you count it. The elderly, in general, 50% of the young for the mean response. And this is a distribution. We have only about 15% of people over the age of 65, and our mean age is about 80, who are really low. The rest are high or the same. We then followed almost 400 of them over 2 years, bleeding them every couple of months. And what we found is we divided them, and their pattern was pretty consistent. If they're a low responder, they pretty much stayed that way. Every so often we'd have a blip. We would have some of our high responders go to mid, but the categorization after we did it about every 2-3 months was pretty stable. And what we found was that if you're a low responder, again, the number's small, about 33% of you would be dead in 2 years. If you're a high responder, only 11% would be dead in 2 years. And it wasn't due to the age, because as you can see, there's not a heck of a lot of difference, and the mid responders are 86. And when we had the medical records of all of these people examined, all the people who died, by one of the infectious disease fellows who was in my lab, who happened to be Dr. Marla Gold, who was our past dean of the School of Public Health. She was a fellow in my lab. She didn't like doing bench work. She loved going through the medical charts, though. We found that there was no correlation. Most of these people just died due to heart disease. So there was no preponderance of infectious diseases in the people that were low responders at all. But when we looked at, so initially we just looked at proliferation, but now cytokines were getting into the picture. People want to know why it's happening. What are the cytokines doing? So the one cytokine, interleukin-2, is known as T-cell proliferation factor. That was its first name. And when we take supernatant from these individuals, we can then quantitate in terms of units, get back to that in a minute, what's there. And there's a decrease, not that much, but there is no change in the interferon, which was interferon gamma, which really kind of stirred up the world. What do you mean there's no change in that? The interleukin-2 is sometimes down and it's localized a lot. Again, proliferation is the tritium. Units per mil. We didn't have ELISAs. So this was all done by bioassays. And you would throw the supernatant from the IL-2 onto T cells and see if they would grow. Same proliferation assay. And we would look for interferon by looking for inhibition of viral infection. So the assays took a lot longer to get done. And again, this was from all the people in the previous chart. So, all that's human. I still have my mouse lab going on, try to put them together. So we said, what the heck? We'll start doing aging studies in mice. But you had to buy them from the National Institute of Aging because none of the suppliers of mice keep them longer than 8 weeks. You might be able to get 12 weeks. And the animals that we're using here, the median lifespan of most mice is 26 months, believe it or not, so a little over 2 years. And we would always get animals at 20 months of age, so that they're kind of comparable to like a 7-year-old person. And what we would see is that just like we looked at proliferation the same way, except you can now see it says number of T cells, because this is a more recent assay, and we now could start counting cells that were proliferating because we now had a label that would show that they're proliferating. But what you can see is that the color didn't come out great. The dotted lines have no con A, and the solid lines have the con A. Con A is a better T cell mitogen in mice. PHA is a better T cell mitogen in people. And you can see that the cells proliferated far better in the young than the elderly. And it's interesting, the CD4 cells were proliferating higher overall than the CD8 cells. I'm going to cut, in order to finish the story, I'm going to kind of go forward from this about 5 years, so that we can look at this more clearly. And we now have a cool dye called CFSE, that when you put it on cells, they all shine bright. All cells pick it up. But as the cell proliferates, the dye is equally distributed into the 2 daughter cells. So what you get is a halving of the intensity of the dye every time the cells proliferate. So you can actually then count in a flow cytometer the percentage of cells in each channel of the intensity, and you can figure out how many cells are there because you know how many have gone into the flow cytometer. And you can also, and that's an internal label, and on the outside you can put the monoclonal antibody to see if it's a 4 or an 8 or other markers. So we now, initially flow cytometer had 2 channels, you can only do 2 stains, and now we're up to like 18. I've never used more than 8. But what we would find is that in the young mice, after 24 hours, both young and old proliferated, and you can see that change, the halving. By 48 hours, the young have only 20% of the cells have not replicated at all. By 72, you're down to 10% and 5%. But you can see in the elderly, there's real slow progress of the cells replicating. So the cells just don't get kicked off when they start it. They're kind of stuck in G0 and don't go anyplace else. All right, but that was going to what we do in people, just kind of general stimulation. That's not who I am. I do viruses. So let's go back to something I know. So I now have found over the course of years another murine leukemia virus that I thought was far cooler. And it was cooler because if you infect the normal young animal, you will have viral titers that you can detect for 4 weeks, and then it goes away. If you then do any immunosuppression in these animals, it's still there, and you can induce a tumor in them. In aged animals, 22 months old when you infect them, basically what you're having is virus titers for 12 weeks. And unfortunately, we can't go out long enough to have them come back again because they're dead. Okay, but all we can tell you is that it's there for a much longer period of time. So we said, okay, what happens with these guys? So if we look at the T cell response by proliferation, and we've gone back to tritium because it was quick and easy, what we found is that the young mounted immune response to their virus, not real high, but pretty well, and the elderly don't. So the reason that we're not getting clearing here is because we're not getting immune response, but if you look back, it actually correlates pretty well, so that you have virus titers in 4 weeks, nothing at 8, and by 2 weeks, you're getting immune response, but by 8, you maxed out. So it takes a while to clear once the virus is there. But even more importantly, the interferon that's induced an immune response is way higher. starting at week 2 in the young than the elderly. So interferon is still important, and we can still get a response. But we've never done viruses in people. So the head of the medicine department, I said I wanted to vaccinate these people. And I really wanted to vaccinate them with hepatitis B because it was a vaccine that was coming out at that time, and a lot of elderly had not been immunized yet. So it was like primary immune response, but the nursing homes or the continuing care retirement communities where most of them live. So these are people who put their money down, live in apartments, and then have the ability for acute care. None of those facilities were requiring hepatitis B at the time. So we couldn't get them to be immunized with hepatitis B. Now they are required to do hepatitis B. So we did what they were recommended to do all the time. And that's how I got into flu. So if I didn't do the elderly, I would never be doing flu. So this is the response that we got in every flu vaccine. You have 3 components. I'm doing antibody titers now. Why am I doing antibody titers? Because that's what everybody uses as the standard for the response to influenza. So even though I love T cells, I would not be able to get my papers published unless I use their standard first before doing something else. But what you can see here is that these are all pre, so we would go to retirement communities. Most of our studies used about 300 people a year. 300 elderly. We normally would only have about 50 young people in the study every year because the responses are much more consistent. But what we would see post-vaccination, we had increase in titers in the young all the time. The elderly, we got an increase, but nowhere near the titer that we'd get in the young. A titer of 40 is, in theory, protective. But another part of our study, we demonstrated that in the elderly, you can have titers up to 640 and still get influenza. So why do we vaccinate you? Because you're not going to get pneumonia and being in the hospital. So your immune response to the elderly is lower, but it's enough to ameliorate the disease so you don't get hospitalized. But you still might get it. Sorry. Don't have to worry about it until you're 65. Any of your parents, remind them that they should you're supposed to get vaccinated all the time now anyway. Although there's something to be said about a really strong primary disease to really generate a good immune response. But once you get older, do not pass up that flu vaccine. It's really important. All right, so this is the same slide I showed you before, with the beginning of what I look for proliferation and interferon gamma and IL-2. But now we looked at those same individuals after they were immunized with influenza. And you can see really dramatic differences in the ability to proliferate in response to influenza-infected cells. And you also have basically no interferon gamma made. So in contrast to the small differences with this really strong kick of a mitogen, when you're really using an antigen, the elderly's response is quite low. Okay, but there's only so much you can do in people. I can't go in and I get their blood, but the cells in the blood aren't normally as reactive as the cells that you would find in any of the lymph node organs. So the spleens and lymph nodes, and there's no way I'm going to be able to do that in people. Shouldn't anyway, but you're not going to do it. So I go back to mice. So I then immunize mice, infect mice with influenza virus. I remove their lungs. I isolate lymphoid cells from the lungs, and then I look at the cytotoxicity assay. But now, also looking at T responsive cells, we now have a new technology called tetramers. So back in the day when I started, we had no idea that the antigen was recommended in the context of the Baker histocompatibility antigen. We had no idea. I actually presented a paper that I was trying to demonstrate how it wasn't defined as closely as they were. It was really cool. So that's in the side paper, this account that was going on at the time. But by now we know T cells recognize an antigen in the context of the Baker histocompatibility complex. So we now, through the marvels of molecular biology, will have the MHC with the antigen, the receptor for the antigen in a 4-pronged reagent that if that T cell can recognize this, it will bind to it and it will light up like a Christmas tree. And so those are tetramers. That's not possible if we didn't have molecular biology, because the antigen that we're recognizing here is only 9 to 11 peptides, amino acids, 9 to 11 amino acids with the MHC around it. So this is a standard assay I always use, and this is now the cool, faster assay that you just need a $200,000 piece of equipment to do it, that's all. So we take these mice, we give them virus, we take out their lungs, we look for virus, and what you can see is a decrease in viral titers. And the adult mice basically have no virus after 7 days. The aged mice have virus through 14 days, through 10 days. But by 14 days, they're all pretty much clear because we used a dose of virus that wouldn't kill them. So we have to titrate it because we don't want the animals to die because we want to check their immune response. So we have to give it a low enough dose so they don't die. And this is what we find by the cytotoxicity assay. The chromium assay that we've used all along is that what you have in the young animal, you have an immune response that goes up, peaks by day 10. Remember, it was around day 7 that you got the big decreases, all gone by day 10. In the elderly, in the aged mice, you get no cytotoxic T cells observable until about day 10, and your peak, which is on day 14, is still statistically lower than what the peak that you get in young. So it's delayed and lowered. But through the flow cytometry, we can then look at exactly what's happening. So these little things over here, NP and HA, every genetic background recognizes a slightly different major epitope on the virus. So the nuclear protein and this particular amino acid sequence is seen in one strain of mice, and the HA is in the bowel mice. So what you do is you take your cells and you mix them in culture for 5 hours with the virus that you're talking about, which is PR8. During that 5 hours, those cells have already, because they came out of the animal, have the marker being activated, which is a CD44. But what happens in those 5 hours, if they're going to proliferate and you're going to see the response stronger. So what you have here is, if young animals, you have about 5% of all your CD8 cells are responding to the virus peptide, and you have only about 0.5% in the elderly. But if you go to BALB/c mice, the response overall is lower, but you still have an incredibly low amount. And what you're doing here is that the peptide is on this axis and the marker for inactivated cells on this axis. So using that type of technology, we find a very similar response. So it doesn't matter if we use kind of the simple assay or the more elaborate assay, we get the same kinetics. So that's, to me, really reassuring that we can then take data that was done years ago and still compare it to things that we have today. I always like to do stuff like that, little extra. And in that same combination that we were just talking about, you put that virus or peptide in the culture, and what you can now have an antibody for IL-2, an antibody for interferon. You've already had the antibody for your CD8 in there. You pull those out, then look for the others, and you can now see that in the cells of the young on day 7, CD8 cells, that the cells 2.5% produce both IL-2 and gamma interferon, and another 9% produce gamma interferon alone. Whereas if you go to the elderly, you only have 1% that had gamma interferon alone and 0.2% that produce both. Again, the immune response is not only decreased numbers, but in decreased cytokine production. But then we're getting even fancier. We now have mice whose all T cell receptors in the animals are specific for influenza peptide. So these are transgenic mice, and so these mice in the BALB/c background represent that HA peptide that I showed you. So why do we care about this? And I can differentiate between this transgenic mouse, which is BALB/c, and the wild type by another marker on the T cell. This thigh marker was actually the first marker we had way back when we were doing T cells versus B cells. But what I can now do is I can take these T cells from the BALB/c mice that are all for the influenza virus and put them into a wild type mice that's old or young and say, is it the T cell that's a problem? Or is it something around the T cell that's a problem? Because when you're putting something in the whole animal, how can you tell? Because the immune system is a series of all interactions. So doing that, so this is, we took these transgenic cells and we put them into aged or young BALB/c mice. Oops. So this is putting them into young and then stimulating with the virus. 23% of these cells that we put in are proliferating and responding to the virus, whereas only 11%. The same young cells put into an old environment are not responding. We also did the reverse, and the old cells have a decreased response, but not as dramatic as the effect of the environment. So what is it? Well, the immune system, it's not the T cell acting by itself. It's like everything else in life. You need a team. And so in order to get that T cell to respond well, you need dendritic cells to present the antigen. So the question is, do we have decreased numbers of dendritic cells in the old environment? And the answer is, yes, we do. But if you take them out and put them together, that the old dendritic cells seem to work perfectly well. So it's not that they're not functioning, we just are a little short. And in subsequent experiments, we would take the virus, mix it with dendritic cells, and inject it into the animal, and we'd get an increased response. Not as high as we wanted, but pretty good. But is there anything else that negatively affects it? So we used to know that there were T cells. Then there are T cells that were CD4s and CD8s. And then we got markers for activation. And now we're finding that there's markers, it used to be called suppressors back in the 80s. They went out of favor. Suppressors don't act anymore. They're now T regulators. And why are they different? The suppressors were CD8 cells. The T regs are CD4 cells. Which almost makes more sense because they're the ones who start the whole process. So why wouldn't you think the regulator would be there early rather than something that doesn't respond until a little bit later? So the markers are, there's CD4 cells with CD25, which is actually the IL-2 receptor, and FOXP3, which kind of has some function, but. So what we find, just looking at the number of these Treg cells, is that as you get old, you get more of them. So you're getting less cells that present antigen and more cells that have the ability to depress the immune response. And when we then took spleens out from young and aged and depleted the Tregs by using the monoclonal antibody to then CD44, what you found is, and then this is proliferation by that change in dye, decreasing in half every time they replicate. So as the cells go this way, they're replicating more. So you can see in young spleens over I think this was 48 hours, 94% of these clone cells, so see those cells that we put in, proliferate. We remove the Tregs, it looks the same. In the elderly, only 46% proliferate, but if you remove the Tregs, you have the same exact response as the young. And what we found was in the flu infection, not only do the elderly mice have more Tregs, they actually go up during the infection and go back down again. So they're bad to begin with, and we just make them worse during the immune response. So What we found over these things is the T cell response decreases with age. The decrease is extrinsic, which means the environment impacts it, as well as intrinsic. There's something in the T cell itself. And the changes that we find in mice, whether we're talking about general assessment or response to viruses, seem to be pretty similar to humans. And so we can use them as a model. Now what I wanted to go, I've been putting things in over the course of my career. So I started with hamsters in 1972, have not gone back to hamsters. Started during my postdoc working on leukemia in mice, continued leukemia in mice, but threw in the role of interferon, which has never quite left. And I didn't include a count of time. One of my favorite models is a melanoma model in mice, where you actually put melanoma cells, you're not going to like to hear this, melanoma cells in the foot pad, and then you resect it as a tumor, so you cut off the foot pad. If you resect it at the right size, you get no metastasis. If you resect it at a little bit bigger, you get 100% metastasis to the lung. And we found if you give interferon before you resect, you can cut down the metastasis by 50%. And my graduate student who worked on that with me was an MD PhD and did his residency and is still at the Mayo Clinic in Rochester, and they've used it as part of therapy, so that was kind of cool. Started in the lab. So that was where I came in the 70s. It was in 1982 that the head of the medicine department got me involved in human aging. I then put it into mice about 87. I also had a study on rats. They live longer, so we can look at the immune response. We didn't do any viruses. We just looked at general response, but we really looked at dietary restriction, which is big in aging. Totally long story, so we can't get there today. But in both mice and human, this impact in influenza has been the focus of my work the primary focus of my work for 10 years. And also I want you to see that when we looked at the technologies that we've used over time for cytotoxicity by T cells, I had to count them visually. We went to chromium release. We went to flow cytometry. And then multiple markers on flow cytometry. And again, the first assay to do this was in '69. The initial identification of T cells, we can only do it under a fluorescent microscope, and again, had to count visually. Count the cells that didn't shine and count the cells that did shine. We then went to the flow cytometer with few colors and multiple, and now we can actually look at antigen-specific cells and incorporate cytokines into those cells as well. And monoclonals were developed in '80. Proliferation is thymidine. Then it was a dye, so it's more friendly to the environment. And now flow cytometry is the way that most people go. Cytokines, bioassay, labor-intensive, to ELISA, to flow cytometry, and to molecular biology. So simultaneously, we'll be looking at RNA for these, as well as some of the proteins. And initially, when I started, there was no such thing as transgenic mice. We didn't know how to do any molecular biology at the time. In fact, there was a moratorium in 1974 not to do any molecular biology for 4 years until they figured out if it was going to destroy the world. So, and these are all, the bottom group are all PhD students who did their work with me, and other people have worked in my lab over time. This is only part, I've had 21 PhD students graduating my lab over the years, but they've worked on multiple projects, and these are the ones that worked on the things I talked about today. So I was trying to get the message across how things change, but your basic passion for what you want can stay with you. And while I was going into aging, the part of viruses and the immune response to them is my soul. And whenever I could, I'd go back to the viruses and the immune system. So once you get attached to something, and if you really like it, you don't have to let it go. You might have to modify it a bit to stay there, but you can still stay there and enjoy it for a very long time. And again, as I started, I would say that Andrea would have done the same thing and been excited forever what she's doing. So I'm glad that I had the ability to share my excitement in just reflecting on Andrea. So thank you.
We've all opened for a few brief questions, so feel free to ask, as is our usual custom in seminar. Oh, you guys were going shy on me?
I know I talked fast, sorry. Yes.
So it looks like you've been in the research game for a while. So naturally, you've been in that period where there's a lot of change in different methodologies and techniques that you mentioned again. Isn't it difficult to learn a new system and relearn a new system as everyone comes up every couple of years?
Yep, it is. And if you'll go through any of my papers, I don't have a lot of molecular biology in it. I actually did a sabbatical on it. And I decided that I really like the cell biology aspect. So I would read and understand the molecular biology. If I wanted to do any of the molecular biology, I would never create a transgenic or try to. I would collaborate with someone else. But if you're already in a field with the cell biology, it's not that difficult. You just, as a new technique that comes up that makes sense for you, you do it. At one point, I wanted to do calcium fluxes in the membranes of old cells because there were some reports about the membranes becoming less fluid. And if you can't actually move the antigen to places you want, you don't get a maximum immune response. Try that technique, and I can never master it. So I said, okay, I'm not going to do that one. So those are the type of things that you decide over time. Which are the things you need? The flow cytometry, the cytokines, they're all essential for what I wanted. Using at least 6 colors at one time was really important for us to really play with and manipulate because we couldn't answer our questions without it. But some other ones, it's like, okay, it would have been nice, but we'll pass for now. Yeah.
So do you feel kind of getting back a little bit more into research more full time? Do you see an opportunity now to kind of go after more high risk, high reward types of approaches? And does that change, do you think, over a scientist's career in the trajectory?
Well, first of all, I will say that I've been dean for 15 years and most deans don't do research at all. So I had a research lab until 2015. And I was continually funded by NIH from 1978 until 2015. But it hasn't been funded for a couple of years. And the sad part about science that I can't do my research with today's prices unless I have about $300,000, $350,000 a year, because each aged mouse is $125. Okay, you buy them from the National Institute on Aging. So at this point, I want to collaborate with people who want to do something with aging, but I'm not planning on doing it myself because it's going to take me a while to get up to a grant that's going to give me $350,000 a year to play with. So I'm going to be a mind for people and maybe some hands. I think I'm still pretty good at it. Do IV injections in mice. One of my fortes, but yeah. Yeah, there are things I would love to do, but it's just not, after being out of the game for like 3.5, 4 years, it's going to be really difficult to get grants that big.
Any other questions? All right, let's thank Dean Marasco. Pointing in the wrong direction, sorry. So at this point, we would like to present our award. So you've heard me speak previously about Andrea and the wonderful person that she was, but Andrea Messina was a vital member of our graduating class of 2014. On May 31st, 2014, Andrea lost her all-too-short battle with AML due to complications from chemotherapy. It was with heavy hearts that the biology senior class of 2014 said goodbye, and we dedicated our 2014 senior seminar in her memory. We will forever remember her smile, her kind spirit, and we will mourn her loss from our biology family as we go forward. Although being here today, we're reminded of our loss, today is really about the celebration of Andrea's life. Though all too short, a life well lived that was full of hope, love, laughter. We are grateful to the Messina family for sharing Andrea with her Drexel family and in establishing this memorial award in her memory. Last year, we honored Andrea with a memorial as a career panel, and we invited back many of her dearest and closest friends. They shared their life and career advice with our graduating class, but the Messina family felt very strongly that they wanted to be doing this on a long-term basis and establish an ongoing memorial award, and they were certain that they wanted it to be given to a graduating senior preparing for that important next stage in their life post-Drexel. Some of our last memories was of Andrea sitting out there, the same as you are today. And so that's why we felt very strongly that we wanted to make sure that this award represented that population. So a general thank you to all the biology students that submitted their application this year. It was fabulous to see what everyone has been doing. We have interacted with so many outstanding students over the years, and actually we've had very few ways to formally recognize those who have made a strong and positive impact. It is our hope that this award will express our gratitude for your passion and your devotion to the Drexel biology community. We also hope that this award can help to inspire the same vibrant enthusiasm for science, life, and learning in future cohorts. We want to inspire others to give back to the department and to the community beyond. These were the values that established Andrea Messina's legacy here. They are the things that help make our large group of focused, hardworking student body into something that begins to resemble a cohesive, intimate community. The staff and faculty are happy to be a part of this community, and we thank each and every one of you for being part of that community and for being so dedicated. Thanks to all of you, again, who applied for the award, telling us in your own individual ways about the things that inspired you as a Drexel student and how you've tried to give back. Today, we'd like to recognize just one winner, but after that, we'd like to share and tell you about some of our 5 finalists who were also very impressive as we looked at the candidates for the award. Each of them has made their own mark here, and we are proud to tell you who they are and how they have made a difference. So at this point, I would like to announce the award for the 2018 Andrea Messina Memorial Award, and our recipient is Stefan Lakhar. Stefan's impact has been palpable during his time at Drexel. He is truly engaged with the people and opportunities that we offer here. He has formed meaningful relationships with many of our students, staff, and faculty. His letter writers described how he simply light up the classroom, and I love that picture. I have hijacked these from the class list. Inspiring his peers and elevating the level of discourse among our faculty and students. Stefan is someone who has worked hard to reach his current academic standing, and he is someone who cares genuinely about learning. In these respects, the committee saw much in Stefan that reminded them of Andrea. People have really appreciated getting to know him and how he has gone out of his way to be friendly and engaging to so many. Stefan has also been very involved through his service to Drexel and to the biology department. He is with the Drexel's Emergency Medical Services. He has been a UNIV 101 peer mentor for the past 3 years and was a valuable person in providing feedback that helped to improve this course. Stefan is also now serving as a TA for Biology 126, where he gets to work with freshmen, and I love the fact that he is on the other side, looking at what it feels like to be in the front of the classroom as he's working with these first-year students in training. He has long been one of our go-to ambassadors for undergraduate recruiting events. The excitement that he shows about biology experience is contagious, and we have little doubt that this has inspired others to join our department here in biology at Drexel, and for many to stick with the major after times of early uncertainty. Enthusiasm, inclusivism, engagement, positivity are all words that come to mind when we think of Stefan. He has been an asset to learning and outreach, and we know that his contagious enthusiasm will carry him far. We would be hard-pressed to find someone who so well exemplifies Andrea's best attributes. So we are simply delighted to announce Stefan as the first winner of the Andrea Messina Memorial Award. We thank him sincerely for all that he has given to us during his time here, and we hope that we have been able to repay him in kind. We wish him well, the best of luck in his future career as a medical professional. I would also like to invite up our other finalist, and I would like to say a few words about each as they meet the Messina family. And again, having hijacked the pictures from your class list, Effie Matthew in her application talked about the professors here who inspired her and about the fun way she made learning fun, including catchy jingles about RAS, RAF, MEEK, ERK pathways. I don't know how that jingle goes, but I would love to hear it. It was clear that she enjoyed being a biology student. Effie's application and letters conveyed a strong sense of someone who has been inspired by one cause during her time as an undergrad, and who has worked to further this cause. This was evidenced through her 4 years of volunteer teaching at Samuel Powell Elementary in Powhatan Village, as part of the American Reads American Counts program. Her letter writers describe Effie as thoughtful, caring, and dedicated. They also recognize her for truly meaningful, impactful work, pointing out how she has worked with Drexel's Lindy Center to formalize the partnership between Drexel and the Powhatan community, which has greatly increased the tutoring services that we offer to this school. On top of this exemplary work, Effie has been a great citizen of the university, playing key roles that have included service as a peer mentor in UNIV 101, as an admissions office student ambassador, and others. We wish Effie the best of luck in her future plans as she earns a master's in public health and to eventually become a physician. Nathia's time at Drexel has been characterized by a long-standing commitment to Jalak, an all-female Drexel dance team that combines Indian and Western styles of music and dance, including genres like Bollywood, jazz, and hip-hop. Nathia has served in the group's president since her junior year and has clearly given her all to leading this group and to inspiring them with her positive attitude. She has also played a role as a leadership consultant through the Office of Campus Engagement at Drexel, where she has worked to expand the opportunities for Drexel students wishing to cultivate their leadership skills. In addition, she has served as TA for Drexel's Civic 101 course, where she was described as being an excellent role model for her younger Drexel peers. In her, they saw someone with meaningful commitments to civic and university engagement. One would hope that her example has inspired others to follow suit. We thank her for giving so much to the Drexel community and wish her luck in her future endeavors as a clinician. Timothy Mott. Timothy has been a very active member of our student community. He served as the founding member and president of Drexel's chapter of Health Guardians of America. This nonprofit organization has a stated goal of reducing risk factors associated with long-term chronic illness. For the leadership, management, and organizational skills shown throughout this work, he earned strong praise from his references. Timothy has also been an active member of the Renewal College Fellowship, which has allowed him to become engaged with the surrounding Philadelphia area community. His warm, positive, welcoming personality has made him a great asset in this role. In our department, he is viewed as a valuable contributor to in-class group activities and was recognized also for showing leadership skills in the classroom. Timothy is also an aspiring physician, so we thank him for being such an engaged member in our Drexel community and wish him the best of luck on his future career path. Jasmine, what is it, Zavi? Jasmine's letter writers describes how she went above and beyond during her co-op at GSK, learning the ins and outs of heart physiology out of genuine curiosity and a drive to become well-versed in the discipline of her co-op research. This passion for learning was a theme in her letters, as was her good-natured, upbeat personality. Jasmine has played a role in our department through her 2 years of peer mentoring in UNIV 101. She volunteers with her church and with the Lymphoma and Leukemia Society, and she has also served as a tutor in general. It's clear that Jasmine has made her mark during her time at Drexel as an undergrad. We are grateful for all that she has done and wish her the best of luck in her future career as a physician. Aditya Patel. Aditya has been very active during his time at Drexel. Among the more notable of his commitments, he was his service to Sigma Phi Epsilon and his role as vice president of that fraternity. Through this engagement, he has shown a commitment to learning by helping to oversee Sigma Phi Epsilon's residential learning community. The numerous classes and outside speaker events he has organized have focused on topics including CPR, alcohol safety, and the prevention of sexual assault. Aditya also earned his EMT license while here at Drexel and has been a member of Drexel Club swim team. Like our other finalists, he too is embarking on a path in medicine. We thank him for his service and we wish him well, the best of luck for his future career. Again, we just want to say how truly awe-inspiring the group has been. We want to thank everybody that has applied. And I would also like to take the time to introduce the committee that was the award committee for this award for Andrea. Karina Gerveshi, which was one of Andrea's dear friends. Steve Zario, her boyfriend, chair of the committee, and the one who has kind of done the yeoman's work. Thank you to Jake Russell. Andrea was doing research in his lab. Karen Kavnik, who was instructor, friend, and mentor to Andrea in so many ways, and especially to the Messina family. Her lovely sister, Sarah, and Debbie have been working with us on the committee, and at this point, I would like to have the family come up and say a few words.
Thanks.
I can hold it if you want.
Oh, yeah, that's good. Okay. Good afternoon. First, I heard a lot about senior seminar from my Andrea, and she would tell me about the good, the bad, and the ugly. And I understand that you have about 5 weeks to go, so I want you to know you can do this. Also, I really do want to express a sincere thanks to the dean. Over the years, just knowing her while Andrea was here has meant an awful lot. And your science today was very interesting. You've done a lot of wonderful things. And I do look forward to hearing and being a part of what you do in the next years ahead of us. We also want to thank the committee. They worked effortlessly. They were just spending so much time and energy to make this happen. To each of the applicants and our finalists, all your essays were impressive, they were strong, and we sincerely felt your heart. We've sincerely felt heart. You know what I mean. Your accomplishments at Drexel in the community were notable and appreciated. We not only want to wish the applicants, finalists, but all of you continued success after graduation. You're sincerely in a very good place. You're surrounded by very good people, and I'm sure that over the years, you've made some very good friends. So right now, I'd like to have you give yourself a hand.
As I said, we would like to thank the Messina family, not only for sharing Andrea with us, but for taking their love and turning it towards this wonderful award for the amazing students within our department. In closing, just in honor of Andrea, I would just like to encourage all of you to live life to the fullest with no regrets, love and be loved, and laugh as much as you can. So thank you for your presence here today, and thank you for sharing with us the memory of Andrea.
Thank you.